The ability to see is one of the most fascinating senses. Although we are able to differentiate between various shapes and colors, the resolution of the eye is limited. In order to display smaller structures, like cells and bacteria, we need tools like the light microscope. With its help, objects of a few micrometers can be displayed. But is it possible to visualize even smaller objects, like the surface of a cell? In this video, we'll present you the microscopic technique with the highest achievable resolution, the atomic force microscopy, short AFM. Due to its resolution of up to 0.1 to 10 nanometers, the imaging of molecules and even single atoms is possible. This can be used to determine the characteristics of a surface or to measure the atomic forces at nanometer scale. Lastly, we'll also introduce a few fields of application of AFM. Let's have a look at the buildup of the AFM, which makes the insight into this nanometer world possible. The main element of the AFM is a cantilever, to which the measuring tip is attached. The cantilever is a flexible beam embedded to a microscopic spring. This sensing element is guided over the sample surface by a piezoelectric scanner at a very small distance. The movement of both, the scanner and the tip, is regulated by a controller. This process is called scanning. Depending on the surface structure of the sample, the cantilever bends varying degrees according to its base. The bending of the cantilever is measured by a laser beam, which is focused on the tip. The positioning of the laser and the tip is enabled by a photo-optical microscope. The reflected beam is captured by a photodetector. The result is a 3D topographical image allowing the determination of the sample's surface properties. In the contact mode, which is mostly used for solid substances, the cantilever is in direct contact with the surface. In cell biology, the non-contact mode is used. Here the tip is not in contact with the sample, but the cantilever deflects due to attractive or repulsive forces between the measuring tip and the surface. Atomic force microscopy can be used to image local surface characteristics with high selectivity, accuracy, and resolution up to atomic level. The measurement is possible in various environments without any sample processing or destruction. Consequently, the AFM is often used to measure interactions between antigen and antibody, misfolding of proteins, or protein-DNA interactions. With the AFM, we are able to image the landscape and the virus-binding events with a cell. We can also analyze single biomolecules on the membrane surface, like proteins, and giving insights into the mechanical behavior and structural properties of single molecules. It is also possible to analyze the nanostructures of the DNA. But how is this possible? We want to show you a whole measuring process of the AFM using the example of study of single biomolecules. It is a molecular stretching experiment. The tip is moving up and down above the cell membrane surface where the proteins are located. Then it is brought into contact with the sample protein. While the tip is moving up, the molecule is stretched. Thereby, deflection and separation are measured and converted into a force separation diagram. In the diagram, the y-axis describes the vertical deflection and retraction of the cantilever in piconewton. The x-axis shows the separation between tip and sample. Consequently, a force profile is received. Steps of force profile: tip away from sample, contact, retraction, adhesive event, attraction, different distance between tip and sample. It is fascinating to make these small molecules visible with the method of AFM. The AFM was a big innovation in cell biology, but it's not the end of progress yet. In the future, the possibility of combining different microscopes and modes might offer an even better resolution and new opportunities for research. We hope you understood the AFM and its application in cell biology now. Thanks for watching this video.